Welcome back to the Deep Dive. Today we're plunging into something pretty fascinating, I think, how plants actually build themselves. It sounds simple, but it's incredibly complex.
It really is. And it's strikingly different from how animals develop, isn't it?
Totally different. Animal development. Mammals especially, you get that basic body plan locked in really early. Cells move around, things slot into place.
Yeah.
It feels very predictable.
Right, almost programmed from that single cell. But plants, they play a different game entirely.
Yeah, flexibility seems key. Adaptive growth, what they call indeterminate growth. Our source material for this deep dive, a chapter from Plant Physiology and Development, really highlights this contrast. Think about like a giant sequoia.
Thousands of years old.
Exactly, just slowly adapting, growing around obstacles, reaching for light, all from one spot.
Compared to, say, Arabidopsis, the little weed that researchers love.
The lab workhorse.
Yeah.
Seed to seed in, what, a couple of months?
Yeah, super fast life cycle. And that difference, it's not just a fun fact. It gets right at their core survival strategies.
Because plants are photosynthetic, right? They're stuck in one place.
Exactly, sedentary. So they have to adjust their growth constantly, optimizing for light, for nutrients right where they land. Animals, being mobile, could evolve a more fixed body plan early on because they could move to find resources.
Okay, so that's our mission then.
Yeah.
Let's unpack this plant strategy. How do they pull off this dynamic, adaptive way of building themselves?
Yeah, we'll dig into the source chapter, pull out the key physiological processes, the developmental stages, the molecular details, which are pretty cool.
And the experiments that showed us this stuff.
Right. The goal is to give you a really solid grasp of how plants construct themselves.
The source mentions the big challenges for biologists studying this. First, just describing it, all the growth, the complexity increasing, how cells divide, expand, differentiate.
And how the environment constantly shapes those processes. It's a moving target.
And then the second challenge, figuring out the mechanisms. What are the genetic blueprints? How do plants follow internal programs, but also respond to, light or water or temperature changes?
And what about the physical parts? Like cell walls, how do they play into it? And regulation, how is this whole dynamic process controlled?
It seems like a lot hinges on this concept of meristems.
Absolutely central. These specialized tissues are the key to that ongoing, flexible growth.
Okay, so let's start with the big picture of plant growth and development. You mentioned the sedentary lifestyle forces this different approach.
Right, their structure is optimized for resource capture from a fixed spot, a relatively rigid anatomy compared to animals, but the growth is flexible.
And growth happens by adding new cells locally from these meristems, not by cells migrating long distances like in animal embryos.
Precisely. Meristems are basically reservoirs of stem cells, undetermined cells that just keep dividing.
That's the source of the flexibility then, this pool of cells ready to become whatever's needed.
That's the idea. It allows for that adaptive growth the source keeps coming back to.
So the chapter outlines 3 main stages in a seed plant's life, right? Figure 17.2 lays it out. First up, embryogenesis.
This is the very beginning. Inside the ovule, which becomes the seed, you go from one cell, the zygote, to a tiny, basic, multicellular structure.
And this stage, unlike later growth, is pretty predictable.
Very predictable, yeah. Highly patterned. It has to be, really, to package everything efficiently into a seed. It lays down the fundamental top-to-bottom axis, the polarity, and establishes the very first meristems. Then it prepares for dormancy.
Okay, so after the embryo is formed, the seed chills out for a bit, dormancy. Then stage 2, vegetative development.
Right. Triggered by the right cues, could be water, temperature, light, even smoke for some species. Germination begins.
And the seedling pops out, initially living off stored food from the seed.
Exactly. Uses those reserves first. Then, once it gets established, figures out light direction, undergoes photomorphogenesis, which we've talked about before. It starts photosynthesizing and really takes off.
And this is where those meristems kick in properly. The shoot apical meristem, the SAM at the top, and the root apical meristem, RAM, at the bottom.
That's right. They drive this stage. And this growth is usually indeterminate. It just keeps going, adapting the plant's form to whatever environment it finds itself in, building stems, leaves, roots.
Until stage 3, reproductive development.
The switch flips. Often triggered by internal factors like the plant reaching a certain size, combined with external cues like day length or temperature changes.
And that's when flowers appear.
Typically, yes. The shoot apical meristem often transitions, changes its job description, basically, becomes a floral meristem, and starts producing flowers instead of leaves, which, leads to the next generation.
Okay, broad sweep. Embryo sets the stage. Vegetative growth builds the body. Reproductive growth makes flowers and seeds. Let's zoom back into embryogenesis because setting up that initial polarity sounds crucial.
It really is. It's where the basic architecture gets laid down. You've got morphogenesis, the shaping of the form, organogenesis, making rudimentary structures like cotyledons, and histogenesis, differentiating the first tissues.
And critically, establishing those apical meristems, SAM and RAM, they're the engines for all the later growth.
Exactly. And prepping for dormancy, getting physiologically ready to wait.
The source mentions differences, like between eudicots and monocots, think Arabidopsis versus rice, but stresses the fundamental processes, especially polarity, are shared. So let's use Arabidopsis as our model.
Sounds good. It starts with the zygote polarizing. Then it divides asymmetrically one small apical cell, one larger basal cell. That first cut is really important.
Then the globular stage.
Right. The apical cell divides, makes a little ball of cells. The early embryo proper, it's radially symmetric at this point, forms an outer layer, the protoderm, the future epidermis.
Then it starts looking less like a ball, the heart stage.
Yeah. Cell divisions get focused, push out two little bumps. Those are the beginnings of the cotyledons, the embryonic leaves. Now it has bilateral symmetry.
Okay, then torpedo stage, just elongation.
Elongation, yeah, and more differentiation. Refining the shapes, starting to see differences between the top and bottom sides of the cotyledons.
And finally, mature stage. Drying out, storing food, going dormant, ready to go.
Yep. And through all this, two fundamental axes get established. The apical basal axis shoot tip to root tip, set up by that very first asymmetric division driven by, differences in the cytoplasm.
And the radial axis.
Center to surface. This defines the concentric layers. Vascular tissues inside, then ground tissue, then epidermis outside.
And that first division is key for cell fate too, right?
Yeah.
Figure 17.5 shows the apical cell makes almost the whole embryo.
Pretty much. Cotyledons, SAM, hypocotyl, the main part of the root, upper ram.
While the basal cell forms the suspensor, that sort of tether.
Right, anchors it. Though the very top cell of the suspensor, the hypophysis, does contribute to the root meristem, forming part of the root cap and the quiescent center.
Okay, now this is where the source really challenges simple ideas. It argues against a strict cell lineage model, like you see in some animals.
Exactly. In plants, it seems position matters more than lineage, where a cell seems to be the main determinant of its fate, not necessarily which cell it came from.
What's the evidence for that?
Well, one thing is just looking at other plant species. The precise patterns of cell division can vary quite a bit. If lineage was everything, you'd expect it to be rigidly conserved.
Okay.
And even in Arabidopsis, where it's pretty regular, fate mapping shows there's still some flexibility. Cells don't always end up exactly where a strict lineage map would predict.
But the killer example is the fast mutant, right?
Oh yeah, FAS is amazing. Figure 17.8 shows it. Cell divisions are all over the place. Expansion is weird. The embryo shape is totally distorted.
So like a mess.
It is a mess. But, and this is the crucial part, it still forms recognizable tissues. Epidermis, cortex, vascular cylinder, and they are in the correct radial positions. Outside, middle, inside.
Wow. So even with total chaos in how the cells divide and grow, the positional information for tissue layering still gets through.
That's what it strongly suggests. Spatial cues seem to override the need for a precise sequence of divisions to get the basic pattern right. It speaks volumes about plant developmental plasticity.
So position-dependent mechanisms. That implies cells need to know where they are and talk to each other, right?
Absolutely. Intercellular communication is key, and early embryos are surprisingly chatty. Figure 17.9 shows experiments using GFP, a fluorescent protein. Even fairly large molecules like that can move between cells early on through the Plasmodesmata, those channels connecting plant cells.
Like an open network.
Initially, yeah, relatively open, but later in development, that movement becomes more restricted. It seems the plant starts gating those channels, controlling the flow of information to create distinct developmental regions.
So how do we figure out the genes controlling all this? Mutants.
Mutants are indispensable, especially ones called seedling defective mutants. They manage to make a seed, but when it germinates, the seedling's organization is all wrong. That tells you the affected gene is crucial for embryonic patterning.
Like Gurk and Fackel.
Yeah, Gurk mutants lack cotyledons in the SAM. The gene deals with lipid synthesis. Fackel affects sterol synthesis and has messed up cotyledons and roots. It points to lipids and sterols being important, maybe for signaling or membrane structure needed for patterning.
And the really key ones seem to be Nom and Monopteros.
Definitely. Non-mutants have severe disruption of that top-to-bottom apical basal axis. The gene, GNOM, is vital for getting transport vesicles to the right place in the cell, specifically for polarized transport.
Aminopterose or amoeba.
MP mutants are missing the basal parts, the root and hypocaudal. MP encodes an auxin response transcription factor, so NOM points to transport, M points to auxin response. Both scream auxin is critical here.
Okay, let's talk auxin. It's a hormone, acts as a mobile signal. A morphogen.
Potentially, yes. A morphogen is something that forms a gradient, and cells respond differently depending on the concentration they sense. Auxin fits that bill. And a key way plants create these gradients is polar auxin transport.
That directional cell-to-cell movement.
Exactly. It's energy-dependent, found in pretty much all plants, and it creates these specific peaks and troughs of auxin during development. Figure 17.11 shows the model.
Right, the chemiosmotic model. Auxin can enter cells passively when it's undissociated, or actively via transporters like AUX1-LAX.
Yep, those AUXLX proteins use the proton gradient across the membrane to bring auxin in. They act like sinks, pulling auxin into the cell.
But the key for directional flow is getting it out again, right? The PIN1 proteins.
Precisely. The pin-OX efflux carriers, named after the pin1 mutant phenotype, which looks like a pinhead because it can't form organs properly. These PIM proteins are stuck in the plasma membrane, but only on one side of the cell.
They're polarly localized.
Exactly. So if a cell puts all its PIN1 proteins on its basal side, it pumps auxin downwards. If it puts them on the apical side, it pumps auxin upwards. Different pins in different tissues, pointing different directions, create complex flow patterns.
PIN1 being crucial for shoot development. And there are other transporters too, like ABCBs.
ABCB transporters also pump auxin out. They might amplify the flow or work with pins to make the directionality stronger, especially in small cells.
And you can mess this up with chemicals, right, like NPA?
Yep, NPA blocks polar auxin transport. And crucially, treating plants with NPA mimics the defects you see in PIN1 mutants. Figure 17.14 shows this. That's strong evidence linking pin-mediated transport to development, particularly organ formation.
How do we actually see these auxin patterns?
With reporter genes. DR5 is a classic. It's an auxin-responsive promoter hooked up to a reporter like GUS or GFP. Where DR5 lights up, auxin signaling is high. There are newer ones too, like DII Venus, which actually gets degraded by auxin, so it shows auxin presence more dynamically.
And you can fuse fluorescent tags directly to the pinin proteins.
Yes. That lets you see exactly where the pin is located on the cell membrane, which tells you the direction of transport. These tools have been revolutionary. You can literally watch auxin microgradients form and shift during development. Figure 17.14 shows how pin end distribution, and thus auxin peaks, change dramatically through embryogenesis stages.
So back to the non-mutant. GNOM protein is needed to get the pins to the right place on the membrane.
That's the connection. GNOM is a GEF involved in vesicle trafficking. It seems essential for targeting the pin proteins correctly. Without it, pins are mislocalized, auxin transport goes haywire, and the apical basal axis fails to form properly.
It's like the delivery system for the auxin pumps breaks down.
Pretty much. Okay, so that's transport. What about the response? That brings us back to Monopterus, MP.
Which is an auxin response factor, an ARF, a transcription factor.
All right, ARFs like MP turn target genes on or off, but only when auxin is around. Normally, they're inhibited by repressor proteins called IAAAUX proteins. One of these is BDL from the bodenlose mutant.
Ah, the baseless mutant.
Exactly. Auxin causes these IAAUX repressors like BDL to be degraded. That degradation frees up the ARF, like MP, to do its job.
So in the bodenlose mutant, the BDL repressor is stable. It doesn't get degraded even when auxin is there.
Correct. The mutant BDL protein sticks to MP and keeps it shut down. And the bodenlose mutant phenotype, missing the basal region, just like the M mutant.
That ties it together beautifully. Auxin flows, directed by pins, which need GNOM, create patterns. High auxin degrades repressors like BDL, freeing ARFs like MP to activate genes needed for, say, basal development.
Precisely. GNOM controls the flow, MP mediates the response to that flow. It's a system linking auxin movement and perception to downstream developmental programs. The source notes some debate about whether it's pure concentration or the direction of flow that matters most, but the link is clear.
Okay, that covers the apical basal axis setup. What about the radial axis, the tissue layers?
Also established in the embryo, around the globular stage, figure 17.15, you get the outer protoderm, future epidermis, the ground tissue underneath, cortex endodermis, and the central procambium, vascular tissue.
And again, the FAST mutant tells us position is key here too, not just lineage.
Right. Even with messed up divisions, it gets epidermis outside, vascular inside. Positional signaling must be specifying these fates.
Are there specific genes for each layer?
Yes. For the epidermis, transcription factors ATML1 and PDF2 are crucial, figure 17.16. They're expressed only in the outermost cell layer very early on. Knock them out, and the epidermis is abnormal. Put them elsewhere, and you can get epidermal features. They seem to lock in that outer identity.
And the vascular tissue in the middle.
The wooden leg, wool gene, is important there. It encodes a cytokinin receptor. Wool mutants have defects in forming both xylem and phloem properly. Figure 17.1 shows cytokinin signaling is involved in getting that radial pattern right.
And fascinatingly, the FAST mutant can partially rescue wool defects.
Yeah, because FAS causes extra cell divisions, it can sometimes accidentally create a cell layer in the right position for phloem development, even if the wall signaling is faulty. Position matters.
Okay, and the classic example for radial patterning is the cortex and endodermis layers involving scarecrow, SCR, and short root, SHR.
Right. Figure 17.18. These mutants fail one specific cell division, so instead of two distinct layers, cortex and endodermis, they just have one layer.
What's cool here is how SHR works. The SHR gene is transcribed only in the central vascular tissue.
But the SHR protein moves. It travels through Plasmodesmata into the adjacent cell layer, the one destined to become the endodermis. Figure 17.19.
So the protein itself is the signal.
Seems so. Once in that layer, SHR protein activates SCR gene expression and also partners with the SCR protein to turn on genes needed for endodermal identity. It's a clear case of a transcription factor moving between cells to specify fate based on position.
Amazing. Okay, so all this intricate patterning in the embryo sets up the basic plan and, crucially, the meristems.
Yes, the engines for post-embryonic indeterminate growth. Meristems are these populations of self-renewing stem cells.
The source highlights the root apical meristem RAM and shoot apical meristem SAM. Similar strategies, different outcomes.
Both maintain a pool of initials, balanced division with differentiation. But the RAM is adapted for growing through soil, SAM for growing in air and making leaves.
Let's look at the RAM first, figure 17.20. It's got that root cap protecting the tip.
Right, sensing gravity, secreting slime. Then the meristematic zone with the dividing initials. Behind that, the elongation zone where cells stretch out massively, pushing the tip forward. And finally, the maturation zone where root hairs appear and cells take on final functions.
And at the very core of the meristematic zone is the quiescent center, the QC, figure 17.21.
Yep, a small group of very slowly dividing cells. It's surrounded by the initials, the actual stem cells that give rise to the different root tissues. The QC and initials are functionally interdependent.
Experiments showed the QC stops the initials differentiating too soon.
Yeah, laser ablation studies. Zap the QC and the initials start dividing weirdly or differentiating. Zap cells next to initials and the initials can change fate. It confirms that signaling based on position maintains the whole structure.
And hormones are key here too. Auxin.
Big time. The QC sits right at an auxin maximum. If you mess with that maximum, you mess with the QC.
And transcription factors respond to that auxin, like plethora, PLT.
Exactly. PLT genes are turned on by high auxin in the QC zone. PLA mutants can't maintain the QC. Artificially expressed PLT elsewhere, you can get ectopic QC-like cells. Auxin provides the positional cue. PLT executes the program.
What about WX5?
WX5 is another crucial transcription factor expressed in the QC. It acts like its counterpart WUS in the shoot. It maintains the stem cell identity of the surrounding initials, prevents them from differentiating. Its expression is also influenced by auxin.
And cytokinin interacts with auxin here too.
Definitely. There's crosstalk, often antagonistic. Figure 17.23 shows how auxin and cytokinin signaling patterns are almost inverse in the early root tip, helping define different domains and regulate QC division. High auxin seems to suppress cytokinin responses in certain areas.
Okay, shifting upwards to the shoot apical meristem, the SAM, figure 17.24. Also maintaining stem cells, but making leaves and stems.
Right, protected by young leaf primordia covering the tip. The SAM itself has structure, zones and layers, figure 17.25.
Zones like the central zone, CZ, with slow-dividing initials, peripheral zone, PZ, where cells divide faster to make organs.
And the rib zone, RZ, underneath, making the inner stem tissues, and then layers based on division plane. The tunica, L1, L2, dividing anticlinally, maintaining surface layers, and the corpus, L3 and below, dividing in multiple planes, adding bulk.
And lineage tracing shows these layers are distinct, but again, identity depends on position.
Yeah, if an L2 cell gets pushed into the L1 layer, it starts behaving like an L1 epidermal cell. Position trumps lineage.
How does the SAM get set up in the embryo? Auxin again?
Auxin transport patterns are critical. Keep ion proteins, especially PI1, direct auxin flow. Early on, auxin is apical. Then it gets redistributed, creating a low auxin zone at the very center where the SAM proper will form, flanked by high auxin peaks where the cotyledons emerge.
Transcription factors orchestrate this. WSCUC STM, figure 17.28.
Key players. WSS, related to WOX5, specifies the stem cell identity in the center. CUC genes are expressed in that boundary zone between the emerging cotyledons and are essential for separating them and allowing the SAM to form. CLUC mutants often have fused cotyledons and no SAM.
And STM.
STM, a not X gene, works with W8S to keep cells proliferating, maintaining the meristem. It's expressed broadly in the SAM center but gets switched off where leaves start forming.
So how does the SAM stay the right size? It's constantly losing cells to differentiation but has to maintain its initials.
Through a really elegant feedback loop involving WS and the clavata CLV genes, figure 17.3 Erie, WS is needed to maintain the stem cells.
But too much WS would make the meristem enormous.
Exactly. So US actually turns on the gene for CLV3, a small peptide signal.
Right.
CLV3 peptide moves a short distance and binds to receptor kinases CLV1 and CLV2 on nearby cells. This CLV signaling pathway then acts to repress WS expression.
Ah, so WS promotes its own inhibitor.
Precisely. It's a negative feedback loop. More WS leads to more CLV3, which leads to less WS. It keeps the stem cell population stable. Classic homeostasis.
That's clever. And NOx genes like STM also help maintain the meristem.
Yeah, partly by regulating hormones, figure 17.31. STM activates cytokinin synthesis, which promotes cell division. It also suppresses GA, gibberellin synthesis within the SAM, and prevents GA from entering from developing leaves because GA tends to promote differentiation, which would destabilize the meristem.
So STM keeps the go signal, cytokinin, on and the stop differentiate signal, GA, off within the meristem core.
A good way to put it. Now, just outside the core, in the peripheral zone, leaves initiate in very specific patterns, phyllotaxy, figure 17.32.
Spiral, opposite world.
Right, and those initiation sites correspond to spots of high auxin accumulation, figure 17.33. Again, it's dynamic auxin transport, mediated by p-ion proteins, creating transient auxin peaks that trigger leaf formation. A new leaf acts as a sink, drawing auxin away and preventing another leaf from forming right next to it immediately.
It's all about auxin flow patterns.
Seems to be a recurring theme. Finally, the source mentions the vascular cambium.
Another meristem running along the stem root, making wood and bark tissues, basically.
Yeah.
Increasing girth.
Right. Different structure, but it also needs to maintain initials. And interestingly, it uses some of the same molecular players, figure 17.34.
Like WYX genes.
Yep, WX4 is important in the cambium initials, and it's also regulated by peptide signals, CLE peptides, specifically CLE41 and CLE44, acting through a receptor kinase called PXY.
So similar to the WUS CLV system in the SAM and WX5 CLE40 in the RAM.
Very similar concept. Peptide receptor knob WX factor.
Okay.
But here's a twist. In the cambium, the CLE4144 signal seems to promote WX4 activity, whereas in the apical meristems, the peptides repress WX activity.
Same toolkit, wired differently.
Exactly. Shows how evolution can repurpose these signaling modules for different developmental contexts.
Okay, let's try to wrap this up. We've journeyed from the embryo to the mature plant's growth engines. The big takeaway seems to be this dynamic, flexible development based on localized stem cells and meristems.
Definitely. We saw how that initial polarity and tissue layering gets set up in the embryo, driven by positional cues and cell communication, with auxin transport via pins, regulated by things like GNO, and auxin response via factors like MP being absolutely central.
And then how the RAM and SAM maintain those crucial stem cell pools using complex networks of transcription factors, PLT, WOX5 in the root, WUS, STM, NA-X in the shoot, all modulated by hormones like auxin and cytokinin. and kept in check by feedback loops like WSCLV.
And these core mechanisms, these peptide receptor transcription factor modules, pop up again in other meristems like the cambium, showing common strategies for maintaining growth potential.
For you listening, understanding this isn't just academic, right? It explains the sheer adaptability of plants, how they can colonize almost anywhere, how a tree adds rings year after year, how leaves unfurl in those beautiful, precise patterns.
Yeah, it's fundamental plant biology. And the source ends with a really interesting point to chew on. It contrasts simple concentration-based responses to auxin with responses that seem tied more to the direction of auxin flow and the cell polarization it causes.
What does that really mean, developmentally? That the shape of the plant, the vector of growth, might be guided more by signal directionality than just signal strength.
It's a subtle but potentially profound difference in thinking about how form arises. How does that dynamic, flow-based system sculpt an organism?
Something to ponder next time you're looking closely at a plant, watching it build itself.
Indeed.
Thanks for joining us for this deep dive.